Hello everyone. Today we are going to see about cell division and cell cycle. First, let's understand what is a cell. A cell is the structural and functional unit of a body, which means that it is the building block of our body. Now, let's understand the terms cell division and cell cycle in detail. First, let's understand what is cell division. Cell division is one of the most fundamental characteristics of life. This is the method which enables life to perpetuate generation after generation. It is a small segment of a large cell cycle. A parent cell divides into two or more daughter cells. The type of cell division in eukaryotes, that is, in the organisms which have defined nucleus, is of two types, mitosis and meiosis, whereas in prokaryotes, that is, who do not have defined nucleus, is binary fission. Cell division is of two types, mitosis, meiosis. First, let's understand mitosis. Mitosis is the cell division in which one parent cell divides into two identical daughter cells. The most important aspect of mitosis is that the same normal chromosome number is maintained at each cell division. That is, it does not get into haploid numbers. The phases of mitosis are chirokinesis and cytokinesis. First, let's understand chirokinesis. Chirokinesis. Chirokinesis of mitosis occurs in four main phases, although each of these phases merges into the next phase, thereby making it a continuous process. Chirokinesis literally means division of nucleus. The four phases of chirokinesis are prophase, metaphase, anaphase, and telophase. First, let's understand prophase. Prophase. Chromatin fibers shorten and condense to form chromosome. Each chromosome contains two chromatids connected at the centromere. The nucleolus and nuclear membrane disappear. The centrosome divides into two centrioles and move towards the opposite poles. Now let's understand by figure. Here we have covered the first point, that is chromosomes have become short and thick. That is, they have become here short and also thick, so they are clearly visible. Next, the two sister chromatins remain attached to each other at a small region called centromere. If this is a chromosome, the two sister chromatids, that is first one is this and second one is this, is attached by a small dot that is known as centromere. The nuclear membrane and the nucleolus disappear. That is, if this was a nuclear membrane, then this would have been disappearing from the between. So, the nucleolus and the nuclear membrane is not distinct and it starts disappearing. The second phase is metaphase. Metaphase. In metaphase, the duplicated chromosomes arrange on the equatorial plane. Each chromosome gets attached to a spindle fiber by its centromere. Now, if we want to understand it clearly, let us understand by the figure. Now here what is written is that each chromosome gets attached to the spindle fiber. That is, if this is the spindle fiber are the lines that are seen here. If this is the spindle fiber and this the part where two part of the chromosomes are attached is called centromere, as we have seen previously, then each chromosome gets attached to the spindle fiber. That is, the centromere of the chromosome gets attached to the spindle fibers. So each chromosome occupies a spindle fiber. This is what it means. Here, the chromosomes also line up in a single line. That is, the chromosome arrange in a single line or vertical line, one behind the other. Now let's go to the third phase, that is anaphase. The third phase of mitosis is anaphase. Anaphase. In anaphase, the centromere attaching the two chromatids divides and the two sister chromatids of each chromosome separate and are drawn apart towards opposite poles. The drawing action is performed by the contraction of spindle fibers. Now, let us understand this by the figure. It means that the chromosome which was attached at the centromere, that is, if this is the centromere and this is the chromosome, so from this they divide into two parts. That is, this is the one part and this is the second part, and they form two sister chromatids at the spindle fiber and also at the opposite poles. They do not go in one line but they come in opposite poles. That is, if lower part of the spindle fiber is this, then the upper part of the chromosome gets attached to this spindle fiber. Let's go to the last phase of the mitosis, that is telophase. But before we move to the telophase, a observation is made here, that is, a furrow starts in the cell membrane at the middle. That is a furrow, that is a gap starts in between only in animal cell in anaphase. That is, the cell gets downwards and the downward cell gets upwards. That is, this both contract each other due to the spindle fiber contraction. Telophase. Each chromatid or the daughter chromosome lengthens, becomes thinner and turns into a network of chromatin threads. Nuclear membrane reappears. Nucleolus reappears in each daughter nucleus. That is, in telophase, two sets of daughter chromosomes reach opposite poles. That is, two sets of daughter chromosomes. That is, if this is one set and this is another set, they reach at the opposite poles. One goes in left, one goes in right. Second, spindle fiber disappear. That is, the spindle fiber that was present in anaphase like this disappears and becomes negligible to see at the poles. Third, chromatins thin out in the form of chromatin fibers. Now, chromatins thin out. Fourth, nuclear membrane is formed. Nuclear membrane, which means that nuclear envelope around the chromatin or daughter cell is formed or it reappears. Fifth, the cleavage furrow starts depending in the animal cells. And last, nucleoli reappear. That is, the cleavage which was also visible in anaphase, but it was not that much deeper, which is in telophase. In telophase, it goes again very much deeper, and the nucleoli reappear. That is, the nucleus which is the present here reappears. So this was the phases of mitosis. Now let's understand cytokinesis. Cytokinesis means division of cytoplasm. Two daughter cells formed after cytokinesis. That is, cleavage furrow depends only in animal cells and separates two daughter cells. That is, the cleavage which was formed like this in telophase, this disappears completely and they both separate each other from the middle. So this is cytokinesis. So if in brief we want to see all the stages of mitosis, here we go. Number 1 is prophase, second is metaphase, third is anaphase, fourth is telophase, and the last is cytokinesis, in which two daughter cells are formed. Now let's go to the cell cycle. What is a cell cycle? Cell cycle is the sequence of events that occur between the cell formation and its division. Cell division is central life of the cell and is essential for the perpetuation of the species, as we have learned earlier. Now, what is cell cycle? Cell cycle is the event or the sequence of event that occurs between the formation of the two cells. Now, cell cycle consists of two phases, that is interphase and mitotic phase or M phase. The interphase is of three types. Interphase itself has three types. Number 1 is G1 phase, second is S phase, and third is G2 phase. Let us understand this in deep. G1 phase. In G1 phase, RNA and proteins are synthesized and the volume of cytoplasm increases. Now, as we have seen that interphase is also known as the resting phase, but the process does not stop here in interphase. Now, G1 phase, RNA and proteins are synthesized and the volume of cytoplasm increases. Mitochondria and chloroplast divide, that is, two organelles have their own DNA. In late 1 G1 phase, that is, G1 phase is of two types, late and early. In late G1 phase, the cell are given two options, whether they want to continue the cell cycle or they want to leave the cell cycle. So if they want to continue the cell cycle, then they must go through the synthesis phase, and if they want to leave the cell cycle, they again go to the different phases and do their own work. So here in late G1 phase, two options are given to the cells. Now, synthesis phase, that is S phase. DNA replication is done. Here cell growth occurs. So DNA replication, which means that more DNA is synthesized and the chromosomes are duplicated. That is the option which was given to the cells in G1 phase, that if they want to continue. So the cells which wanted to continue go in S phase and their DNA is replicated, which means that DNA is synthesized and chromosomes are duplicated. That if this is one chromosome, then another of its own type is duplicated. Now the last phase, that is G2 phase, that is second growth phase, is occurring. So in this, preparation of mitosis is done. So this is the shorter growth phase in which RNA and proteins necessary for cell division continue to synthesize. So this is a cell division in which it is of shorter length than the G1 phase and synthesis phase. It is shorter because only RNA and proteins necessary for cell division continue to be synthesized. So this is what we have seen in today's session. So hope you have understood. Thank you.